Hello, my name is Mohaimin and I'm here to explain what gene therapy is. But first, let's review what genes are. Genes are the blueprint or instruction manual for all living things, and they can be found in each of your cells. Cells are able to go to work using this blueprint. Everything from the color and the texture of your hair to your height and how your body metabolizes protein in the food you eat is influenced by your genes. Your genetic information is carried through genes, which are formed from your DNA. A change in the gene, often known as mutation, can occur from the original copy. This mutation or defect in the gene prevents the gene from functioning normally. Mutation can occur as a result of biochemical processes within the cell, external factors, or what we inherited from our parents. Genes that aren't functioning properly can cause major problems. Scientists are attempting to develop solutions to these problems. To address a variety of health conditions, they can replace, add, or turn on or off genes. PKU is caused by a mutation in the PAH gene, which prevents persons with a condition from metabolizing the amino acid phenylalanine or PHE. PHE is derived from protein in our diet and serves as a variety of functions, including assisting regular brain function. If the body is unable to correctly absorb PHE, it builds up and acts as toxin, causing a variety of brain issues, including PKU fog. In gene therapy, researchers can replace a dominant gene with a healthy one, or even switch off a problematic gene. Scientists are working on a completely new PAH gene to treat PKU. The PAH gene is created in a laboratory and then transplanted into the body to replace the faulty PAH gene. This new PAH gene will instruct cells to produce a protein that will process, be, and protect the brain. But the new gene must first find a way into the cell. Scientists utilize viruses known as vectors, which are similar to small delivery trucks, to carry the repair gene into your body. But don't you worry, scientists make a virus safe by removing all the stuff that can make you sick. Then the delivery truck, also known as vector, carries the repair gene into the cell. Once the gene moves into its new home, your body will be able to start making PAH enzymes that do what they're supposed to process, the PHE and protein, and keep your brain healthy.
DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, where it is called nuclear DNA, but a small amount of DNA can also be found in mitochondria, where it is called mitochondrial DNA or mtDNA. Mitochondria are structures within the cells that convert the energy from food into a form that cells can use. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. The order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. DNA bases pair up with each other, A with T and G, to form units called base pairs. Each base is attached to a sugar molecule and phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in 2 long strands that form a spiral called a double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequences of base. This is critical when cells divide because each new cell needs to have an exact copy of the DNA present in the old.
So, RNA or ribonucleic acid is mainly composed of nucleic acid, and is involved in many functions within the cells. One of its functions is that it facilitates in the translation of DNA into proteins. So, RNA has three forms, which includes the following. mRNA or messenger RNA. This type of RNA function by transferring the genetic material into the ribosomes and pass the instructions about the type of protein. tRNA or transfer RNA is responsible for choosing the correct protein or the amino acids required by the body into turn helping the ribosomes. It also forms a link between the mRNA and the amino acids, rRNA or ribosomal RNA. So, what are the examples of RNA? For examples, suppose you are a chef and you want to get a recipe from a cookbook. Details of amino acids from a library, ribosomes, but you are not allowed to borrow the cookbook and bring it outside the library. So just like the tRNA, a librarian will lead you to the right cookbook and make you a photocopy of it like mRNA. That's all.
My topic today is about chromosomes. I know that some of us asking or thinking, what is chromosomes? So, chromosomes are condensed DNA, compacted and located in the nucleus of cells, which carries the genetic information of an organism. Humans have 22 chromosomes pairs called autosomes, and the 23rd are the sex chromosomes. These differ between males and females. Among many organisms that have separated sexes, there are two basic types, the autosomes and the sex chromosomes. An autosomes is any of the numbered chromosomes, as opposed to the sex chromosomes. Autosomes are numbered roughly in relation to their sizes. A sex chromosome is a type of chromosomes that participate in sex determination. Human and most mammals have two sex chromosomes, the X and Y. Females have two X chromosomes and their cells, while the males have both X and Y chromosomes and their cells.
NHEJ is defined as non-homologous end joining, which refers to a type of DNA repair system that directly joins two broken strands of DNA. This repair system is useful if you want to create knockout genes, meaning that the gene is inoperative due to premature stop codons being formed. However, this method of repair is also prone to errors and mutations. To simplify things, let's look at this animation. DNA plays a critical role, yet it is also susceptible to exogenous forces such as ionizing radiation and X-rays, as well as genus factors such as replication mistakes, can cause double-strand breaks. Within seconds of forming a double-strand break, the Ku heterodimer recognizes the break and binds to the DNA ends. Ku's high affinity for DNA and abundance in the cell allows for its fast binding. It recruits the DNA protein kinase catalytic subunit. This relocates inwards so it can access the broken ends. Together, they form a synaptic complex called DNA PK, which bridges the broken ends together. The nucleus artemis is sometimes recruited to process these ends, removing any damaged or incompatible nucleotides. The process has little to no end processing. However, a small subset of bricks requires it for accurate repair. The structurally similar proteins are recruited, the XRCC4 and XLF. Their heads contain highly conserved regions which interact, creating a scaffold that forms a bridge between the DNA strands. The XRCC4 and XLF dimers alternate, forming a long-grooved superhelical bundle of filaments. The animator leave one for simplicity. This bundle aligns the end of the ligation. Ligase 4 is recruited in a complex with a towel of XRCC4. It interacts with the two broken ends, undergoing a conformational change to encircle the DNA. Ligase 4 modifies the ends, ultimately forming a phosphodiester bond, like eating the ends together. Non-homologous end joining is error-prone due to its flexibility to directly ligate ends regardless of sequence and without the need of a homologous template strand.
HDR is defined as homology-directed repair, which is a type of DNA repair system that make use of a homologous template to generate the missing parts of a cut sequence. This is more commonly used than NHEJ and is very useful for inserting the corrected DNA sequence or making very small specific changes in the base pairs. This method is seen to have fewer mutations and nearly error-free, or Gene Therapy Net 2018. So, NHEJ is a pathway that repairs double-strand breaks, and that is referred to non-homologous because the break ends are directly ligated. On the other hand, the homology-directed repair is the state of having the same relation or similar position or structure, only to generate the missing parts of a cut sequence. This sequence is commonly used than the NHEJ, and it is more incorrect method. That's all, sir. So if you compare the two sequence or method of DNA, HDR is more appropriate.
ZFNs are artificial restriction enzymes that consist of zinc finger repeats, DNA binding domain, and type 2 endonuclease FOC1 DNA cleavage domain. The binding domain has around 3 to 6 zinc finger repeats, where each zinc finger can recognize 9 to 18 base pairs. ZFNs do have limitations, such as the need to know the exact zinc finger repeats of the target DNA sequence. If the zinc fingers are not specific enough to identify the target sequence, off-target cleavage can occur and cause unintended mutations in different genomes. Zinc finger nucleases are useful to manipulate the genomes of many plants and animals, including Arabidopsis, tobacco, soybean, corn, Drosophila melanogaster elegans, Platinaris dumerili, sea urchin, silkworm, zebrafish, frogs, mice, rats, rabbits, pigs, cattle, and various types of mammalian cells. Zinc finger nucleases have also been used in a mouse model of hemophilia, and a clinical trial found CD4 plus human T cells with the CCR5 gene disrupted by zinc finger nucleases to be safe as a potential treatment for HIV/AIDS. ZFNs are also used to create a new generation of genetic disease models called isogenic human disease model.
Talent are similar to the ZFN. In their cleavage domains, both use FOC1 endonuclease to cleave DNA. However, the main difference is that talents make use of a highly conserved repeated sequence of 33 to 34 amino acids. This offers better matching opportunity to find the targeted sequence. Additionally, it has a divergent on the 12th and 13th amino acid known as the repeat variable diresidue or RVD, which has a strong correlation with specific nucleotide recognitions. Talents are also usually transferred as a template standard, which remove the possibility of integrating into the target cell and also increases the level of HDR. By the way, the meaning of talents is transcription activator like effector nuclease. For example, talents were used to disrupt the LGM locus in the rat and to create a heritable mutation that eliminates LGM functions. For this titration of specifically designed, talents were microinjected either as DNA or RNA into one-cell rat embryos, similar to what has been described for ZFN technology, of which a proportion, DNA 9.5% and RNA is 58%, showed subsequent alteration to the LGM locus. LGM mutation frequency was a function of talents, thus as was the rate of biologically modified rats, which were generated by RNA but not DNA. Injections only. Genetically modified rats were then bred with wild-type like control rats, and the resulting F1 generation was checked for mutant alleles using PCR. This procedure established talents technology as a valid tool for the generation of in vivo gene knockouts in rats.
The CRISPR, specifically the CRISPR-Cas9, is the most modern gene editing tool and the one with the most potential. This tool is far better than previous editing tool in terms of precision, efficiency, and flexibility. Unlike ZFNs and talents, the CRISPR-Cas9 system is based on a prokaryotic immune system. The binding domain, CRISPR, is based on short repetitions of base sequences. Specific sets of genes are associated with the CRISPR repeats, which is why they were termed as Cas, short for CRISPR associated. These genes coded for helicase or nuclease proteins, which can cut or unwind DNA. The best known and most commonly used Cas enzymes are the Cas9, which came from Streptococcus pyogenes. The CRISPR-Cas9 system is composed of three main elements: the Cas9, CRISPR RNA or the crRNA, and transactivating crRNA or the tracRNA. The crRNA serves as the guide RNA of Cas9 to find the target section along the host DNA. The tracRNA binds to the crRNA and Cas9, which forms the active complex. A DNA repair template can also be added to be used by the HDR system. In the laboratory, researchers regularly use CRISPR to alter genes in plant, bacteria, and animal models. If you knock out a particular gene in lab mice, you can directly observe what traits or behaviors are affected. This is how a lot of genetic research has been done for decades, but CRISPR makes these studies cheaper, faster, and more reliable. There are also clinical trials using CRISPR to treat several types of diseases and disorders. For example, at the University of Pennsylvania, researchers are using CRISPR to potentially treat multiple myeloma, a cancer of the blood and bone marrow. They have harvested cells from bone marrow and edited the T cells, part of the immune system, to more accurately target cancer cells before putting them back into the body.
The same technique has been used for sarcoma, a similar cancer. We don't have any result yet, but there is a lot of interest in this approach. SPR-Cas9 is also being used to develop treatments for diseases like sickle cell anemia. In sickle cell anemia, having two bad copies of the beta globin causes severe symptoms, while having only one bad copy produces far fewer symptoms. So researchers have harvested blood cells, run the CRISPR system to repair the one bad copy, and reintroduce the fixed cell back into the bloodstream. These techniques are clever, and they could have amazing ramifications for patients.